Hi, my name's Amelia Gavlik. I did intensive research for about a week, studied Hank Green from Crash Course, bought this sweater, and even went to space camp when I was 12, so that pretty much qualifies me to deliver this TED Talk. Let's begin. The first manned mission to orbit Mars has officially been scheduled for the 2030s. Orbiting a foreign planet equipped with some of the best technology mankind has to offer is one thing, but what happens when we start to talk about inhabiting other planets, establishing new civilizations, venturing out into the unknown, not knowing how long our journey will take, or what challenges we'll have along the way? Well, unfortunately, we are not Iron Man, and no matter how advanced NASA's rocket designs or space colonization strategies are, we still face a huge hurdle in space exploration: the fact that we're only human. Luckily, that is where CRISPR comes into play. Putting it simply, our genetic coding is rubbish in space. Shocker, I know. Here are the main concerns. The massive amounts of radiation in space are incomparable to the levels of radiation our astronauts are exposed to now. You see, the Earth's magnetic field deflects radiation particles traveling at Earth, creating concentrated regions of radiation around our atmosphere called Van Allen belts. So it keeps us, and even our astronauts aboard the ISS, safe. But further away from Earth, where we're exposed to gamma rays and solar flares, we're toast. No, literally, it would fry our cells. From studying the use of radiation therapy to treat cancer, we know that radiation damages a cancerous cell's DNA, thus eventually killing the cell entirely. Now, you may be thinking, Amelia, how does that information help us in space? I don't know much. But luckily, Christina Koch and Nick Hogg do. They are astronauts aboard the ISS who successfully used CRISPR to modify the yeast genome to mimic radiation-caused damage to human DNA. The hopes are that CRISPR can be used to undo that damage and repair these double-strand breaks in human DNA, protecting astronauts from increased risk of cancer and decreased immune and cognitive function. The plan for these little cell-repairing robot genes is to turn them into a drug, which is probably the easiest method to go about, considering we can't just go right in and fix every damaged cell in the human body by hand. But hey, there's another alternative, and it's large, and it's gray, and it has a trunk. It's an elephant. See, elephants have P53, which is a gene involved in preventing cancer, which is why elephants rarely ever get cancer. CRISPR may allow us to knock some of those P53 genes from elephants into human DNA, helping us out with our whole effects of cosmic radiation dilemma. Now, this radiation stuff is pretty cool and all, but let's talk about the other thing that CRISPR has to help us out with: microgravity, which aboard a spacecraft is practically no gravity at all. When being in this unnatural state of zero gravity for too long, we are at risk of muscle atrophy, bone density loss, and suppression of immune function. So far, astronauts have been under a strict exercise schedule, roughly 2 hours a day, with all the adapted workout equipment aboard the ISS to maintain their physical shape. However, physical exercise may not be sufficiently counteractive. The unnatural conditions of microgravity for too long have inevitable long-term effects. But what if CRISPR could engineer stronger bones and more muscle mass for the human body? Well, as suggested by Yale's genetic researchers, possibly taking genes from our closest and much stronger relatives, chimpanzees. Chimpanzees have up to 7 times as much muscle mass as the average human, which would clearly come in handy in our genetic coding. In CRISPR, or any type of gene editing really, there are 2 types of edits: a knockout and a knock-in. A knockout would be removing a gene that carried a disease, such as Alzheimer's or Parkinson's, basically anything we don't want being carried genetically. A knock-in would be any enhancement made to our genetics, like more muscle, for example. Another more reasonable knock-in would be the gene EPAS1, which is a gene commonly carried by Tibetan people that lets you get by with less oxygen. In case you have not been yet wowed by the magic that is CRISPR, what if I told you scientists want to make humans prototrophic, as in being self-sufficient in producing certain nutrients? Well, it would require a combination of over 250 brand new genes per human cell. So whilst we're quite a ways away from that, it's still pretty cool to think about. As far as nutrients go, CRISPR is currently being tested in plant studies in many private labs around the world to see if we can engineer new edible plants that can grow under space conditions, such as plants that don't rely on much nutritious soil or needing gravity to spread their roots. See? CRISPR is cool and delicious. NASA's Human Research Program began funding gene engineering research this year, early January. Gene enhancement through the CRISPR method is set to be available to astronauts within the next 20 to 30 years, if we can reach an agreement in ethical dilemmas. See, not a lot of people agree with introducing CRISPR to the world. In fact, practicing germline therapy is actually legally banned in many areas. For now, CRISPR will most likely be limited to medically beneficial treatments, such as disease prevention or pain stopping, instead of enhancements. But even that is a step in the right direction to helping our future space voyaging superhumans. As Cornell University's Department of Biophysics Christopher Mason says, "You can't send someone to another planet without genetically protecting them if you are able to." That is plenty enough of smart person talk for me today, and that'll wrap up my TED Talk on how CRISPR can help us expand our horizons in space exploration. I genuinely thank you for listening. I'll let myself out.